Okay, we're going to get into Chapter 12 today, which is the lymphatic system and your body defenses, which is your immune system. This is a super interesting chapter, but it is a lot of difficult material. So this is one of those chapters you're probably going to have to go back and listen to a couple of times to fully understand it. And it's very important because this is something you'll be dealing with on a daily basis. All right, now, because the chapter is so large, I will be cutting it into two recordings so that you're not overwhelmed all at one time. Make sure that you go back and you cover both recordings. Alright, as with the past chapters, there are a list of objectives. You want to make sure after listening to both recordings and reading your textbook that you come back and answer all these objectives. That way you know that you've gotten what I feel is important for this chapter. Okay, so this chapter starts off with the lymphatic system. There are two major components. You have the lymphatic vessels, which will transport escaped fluids back to the blood, and then lymphoid organs and tissues, which house the immune cells, which are your phagocytic cells and your lymphocytes. And they filter lymph before it reaches to the heart. All right, so we just talked about the lymphatic vessels are transporting this escaped fluid back to the blood. So where did we get this escaped fluid? All right, well, you talked about this back in the blood vessel chapter, but we're going to refresh your memory. All right, so remember, you've got arteries lead to arterioles, which then feed the capillary bed. And then venules drain the capillary bed. So when you're in the capillary bed, that blood sitting in the capillary, remember you have hydrostatic pressure that pushes all the fluid out of the capillary bed. And the only things that remain in the capillary bed are your red blood cells and your large proteins. That fluid that leaves the capillary, which again is most of your plasma and all that good stuff, goes into the tissues and then, of course, delivers the oxygen and nutrients. Then at the venule end, now you're going to pull that fluid back in with high osmotic pressure. Those proteins that we left in the capillary bed are going to act like a pulling force, and they're going to pull that fluid back into the capillary bed, which now should be full of carbon dioxide and metabolic waste. Now, as with everything in life, we don't get 100% of that fluid back. We get about 80%. So 20% remains in the tissues as interstitial fluid, which is just the fluid in between all the cells in the tissues. So that 20% we cannot leave in the tissues, or eventually you're going to have massive edema where the tissues swell, not to mention that you'll lose blood volume because you're not getting the fluid back in the bloodstream. So that fluid has to be picked back up, that escaped fluid, and that is what your lymphatic vessels do. Now once that fluid enters the lymphatic vessel, now we can call it lymph. All right, so lymph capillaries weave between the tissue cells and the blood capillaries. Every single place there is a blood capillary, there is also a lymph capillary, trying to quickly pick back up that escaped fluid so it's not left in the tissues for long at all. Now, lymph flows one way towards the heart, where blood didn't. Remember, arteries took it away from the heart and then veins brought it back. Lymph doesn't work that way. It always goes toward the heart. Now, the lymph capillaries are extremely permeable, and that's due to those thin endothelial cells that are going to overlap and form flaps, which kind of act like mini valves. Now, the flaps are open when the pressure is greater in the surrounding tissue. That allows the fluid to be drained from the tissue into that capillary bed. Now, the flaps close when the pressure is higher in the lymph capillary. That way they can hurry up and close it so it doesn't seep back out into the tissues. Now, your large bacteria, viruses, cancer cells, these can all enter now that the flaps are open. If the valves stay closed, the high pressure in the tissues again would cause edema. All right, so here you can see in the blue and red, you can see what an actual blood capillary looks like. And then the green is the lymph capillary. So you can see that they're interweaved together, trying to quickly pick up that escaped fluid. This is an illustration of the valves and the flaps we were just talking about. So again, the green is the lymph capillary. So those little valves or flaps are going to open when the pressure is higher outside, thus in the tissues, and then that fluid, once the flaps open, will drain the tissues, and all that escaped fluid will end up back in the capillary bed. Then they'll close the flap as soon as the capillary bed is full. Okay, so what do we do with all this lymph now that we've got it? Well, it started in the lymph capillaries. Then from there, it's going to go to the lymphatic collecting vessels, which are then, obviously they're going to have valves and they're going to have some nodes in there. And then it breaks into the right lymphatic duct, which drains the right arm, the head, and the right side of the thorax. The left lymphatic duct does the remaining 3/4 of the body. And it's a weird setup. You'll get to see it on the picture in the next slide. Anyways, both the left and the right lymphatic duct are then emptied into the subclavian vein. Because remember, we have to filter all of this escaped fluid, but eventually it's got to get back into the bloodstream, or you're going to bleed out. There's no blood volume left. So the subclavian vein is where all that hopefully now cleaned lymph will enter back into the bloodstream. All right, here in this illustration, you got to kind of pay attention. It's not great coloring, but if you look at your patient's right hand side there and the pale kind of mint green, they're showing you what the right lymphatic duct picks up. Again, it's not very much. It's the right arm, the right side of the head, and the right side of the thorax. The left ends up doing the rest of the body. Now, you can also see all the lymphatic vessels and the nodes, and you can see it's a very complex network. All right, these lymphatic vessels are very similar to veins. Not at all like arteries, but similar to veins in the fact that they have thin walls. Their larger vessels are going to have valves, just like veins did, and they're held under low pressure, and they have no pumps, so they use the same mechanisms as veins did to milk the lymph back up. So they milk this lymph by surrounding muscle contractions and breathing. Just simply by you breathing in and out, those ribs are constantly moved, and that will help milk up the lymph as well. Now, you've got smooth muscle, and the larger vessel walls can contract, and that can aid transport. And then you've got something new that veins didn't have. You'll have the pulsating of nearby arteries will also help milk that lymph back up. Lymph nodes. All right, these are filtration units that are clustered all along the lymphatic vessels. However, you have large clusters of them in the inguinal, axillary, and cervical regions. But again, they're found everywhere. They help protect the body by removing foreign material like bacteria, tumor cells, anything your body thinks bad. And they house immune cells, your macrophages, which will destroy bacteria and viruses, and then your lymphocytes, which will produce antibodies and help fight viruses and cancer cells and all kinds of things. Now, the swelling of lymph nodes during infection is due to the trapping of the particles, the bacteria, the viruses, and it kind of gets a little overwhelmed, and that's where you get the swelling, which then also typically causes pain because that swelling pushes on the pain receptors. So let's look at the lymph node a little more closely. Now, it's surrounded by a fibrous capsule, and it's made of reticular connective tissue. Now, the cortex is the outer part. Remember, the cortex is always the outer part. And this collects lymphocytes in the follicles. Then the medulla is the middle part where the macrophages sit. This is showing you the inside of what it looks like under a microscope. You can see macrophages and lymphocytes and the reticular cells. All right, so lymph flow. Lymph enters through the afferent lymphatic vessels. Afferent is always going into something. Now, you talked about it back into the nervous system. We'll talk about it now. You'll also have afferent and efferent when dealing with the urinary system. So it's something that you should be getting more familiar with by this point. So afferent is leading into the lymph node. Then it flows through the sinuses, and exits at the hilus, which is just the branch, by efferent lymphatic vessels. Now, slow flow through the node is due to more afferent than efferent. So what does that mean? That means you have more vessels bringing in lymph than are taking lymph out. That causes almost like a line at the exit. You've got to slow things down, which is very important because that allows the lymphocytes and macrophages to filter and clean and do their job. Now, lymph flows through several lymph nodes before the cleaning is complete. So it's not just, I'm going to run in, get my car washed, and I'm good. You got to go through several nodes to get completely clean. Swollen lymph nodes are called buboes. This is where they are overwhelmed by large numbers of bacteria and viruses. This causes inflammation and tenderness. Now, this is also where we got the name of the bubonic plague, because with the bubonic plague, you had swollen buboes. So they called it the buboe plague, which then led to bubonic plague. Now, lymph nodes can also be secondary cancer sites. Some cancers use the lymphatic system to spread, and that's just bad. Now, swollen glands are usually painless if it's a cancer site, making them very distinguishable from normal inflamed glands. So basically, if you have a swollen lymph node and it doesn't hurt, you need to go to the doctor to get it checked out, because that could easily be a secondary cancer site. All right, now we're going to be moving on into some of these other lymphoid organs like tonsils and thymus and spleen, appendixes, and pyre patches. So we're going to start with the spleen. This is a unique lymph organ in the fact that it filters blood, not lymph, which is, again, kind of weird. But anyway, so it filters blood of bacteria, viruses, and debris. So it's part of the immune system because it's doing what the immune system does. It's just filtering blood, not lymph. It's found on the left side of the abdomen. Now remember, it also destroys and stores the breakdown of old red blood cells, because we want to recycle everything in the body. You know, yeah, you've got an old red blood cell, but the iron's still good, so we're going to keep it. It also acts as a blood reservoir. It can hold up to 25% of your blood at any time, which also is why when you have a spleen injury, it's a very bloody injury due to it being a blood reservoir. Now, it also produces lymphocytes in adults and red blood cells in the fetus. A splenectomy is a removal of the spleen when hemorrhaging. Now, the incidences of us removing spleens has gone down 40% because we now believe that it can repair itself. Okay, that's a huge deal. If it can fix itself, we definitely want it to stay there, because if we take it out, then the liver now has to take over its functions. The liver in and of itself has over 500 functions, which are all vital to survival. You really don't want a busy organ like that having to take responsibility for another organ. It's going to become tired, the toxins will build up, then the kidneys will be the second one to come. It's just a downward spiral. Now, we specifically rarely remove them in children, and that's because we believe it has something to do with the maturing of the immune system, helping the immune system to realize what's good and bad and programming and all that good stuff. So if we do remove it in children, they tend to get sick more easily, more frequently, and then have problems with the immune system as they age as well. The thymus gland. This has peak function during childhood, and then the older we get, the less it functions and eventually kind of shrivels up and goes away. Now, what it does is it produces thymosin and thymopoiens that function to program lymphocytes, your T and B cells, and it's a site of T cell maturation. Now, what does that mean if it kind of shrinks up the older we get? Does that mean that when you get old, you don't program T and B cells anymore? You do. You just program them in different areas. That's all. All right, some of the other lymphoid organs, you have MALT, mucosa-associated lymphatic tissue. This helps protect the upper respiratory and digestive systems. You have tonsils, which are small masses of tissue that ring the throat. They help trap foreign pathogens entering the throat. Tonsillitis is red and swollen due to excess bacteria. And then enlarged adenoids can also lead to difficulty in breathing. Now, the adenoids are found in the back, and we'll be talking about a little bit more about tonsillitis and adenoids when you get to the illustration on the next slide. Now, Peyer's patches, these are found in the walls of the small intestines. They prevent foreign substances from entering the intestinal wall. And you'll talk more about them too when you hit the digestion chapters. All right, here you can see a couple of versions of some tonsillitis. In the upper left-hand corner, you're seeing bacterial growth. That's what all that white fuzzy stuff is. Now, that's obviously going to be a problem, right? But the tonsils are doing what they should be doing. They're swelling full of bacteria and they're trying to fight it. Now, if you look to the bottom right, they're swollen, but there seems to be no bacteria or anything going around. So what's the deal? All right, well, got to go back. With tonsils, we're cyclical. When I was a kid, they took everybody's tonsils. If they gave you any problem at all, they just removed them. We did a tonsillectomy. And then we went through a period where we're like, oh, no, don't take them out. Just leave them there. They'll figure it out. They'll work themselves out. Leave them. And now we're kind of back into an age where we're really approaching this at a much more responsible level. And we're looking at them on individual cases. Because what we do find is that some patients have overactive tonsils. So what does that mean? Well, that means they become super inflamed and exaggerated and swollen for little to no reasons whatsoever. And then we also have some children that, they'll have perfectly normal tonsils, and then for whatever reason, those tonsils will just start growing, not swelling, growing, and they'll take up the entire back of the throat, which then leads to major problems, breathing, snoring, sleep apnea, lots of problems. So we can easily remove those tonsils if that is a problem. Some tonsils like to hold on to bacteria as well and not release them. So then you keep getting these chronic reinfections because they're never being cleared of the tonsils. Now, which of the following filter lymph? You have A, spleen, B, lymph node, C, lymphatic vessels, or D, all the above. The answer would be B, lymph nodes. Remember, spleens filter blood, not lymph. Lymphatic vessels aren't filtering anything, they're just carrying it. Lymph flows slowly through lymph nodes due to A, greater number of afferent than efferent vessels, B, greater number of efferent than afferent vessels, C, dense connective tissue, or D, valves preventing it from leaving. The answer would be A, greater number of afferent vessels versus efferent. Again, you got more entrances than exits, and that caused the flow to slow down. All right, now we're going to be moving on into the body defense portion of this chapter, the immune system. So we're going to get some basic terms to start you off. A pathogen is any disease-causing microorganism. An antigen, remember, is one of those glycoproteins. It's a protein found on the cell surface. Now, they can be perfectly normal for you, which then we call as a self-antigen. Means that your body doesn't see it as a threat. It's on your normal cell. You're good to go. However, non-self antigens typically mean that there is some type of foreign substance. Your body does think that's a threat, and then we'll try to kill it. But we're going to talk more about those later. So self-antigen means it's one of your own cells, your own protein, you're good to go. Non-self means it didn't come from your body. It could be a virus, a bacteria, could be a transplant, Doesn't have to be a bad bacteria or virus, but either way, it didn't come from your body. Now, an antibody is a protein that tags a foreign substance, and it doesn't actually kill it, it kind of tags it so that another cell can come in and kill it. It's kind of like the trees in the forest. You see some of them have X's, and those are the ones that are supposed to be cut down. It's the same type of thing. Antibodies basically mark a bad cell with an X. All right, your body defenses can be broken down into two groups, nonspecific and specific. Your nonspecific defense system will prevent entry and spread of microorganisms. It responds immediately to all foreign substances. You have intact skin, mucous membranes, inflammatory response, proteins. There are a ton of things that are considered nonspecific. The key thing here is that it's literally not specific. It means it's not picky and choosy about what it's going to kill. It'll kill anything it thinks is bad, and it does it immediately. Now, at the bottom, you can see there's a little organization chart, and this will, again, just kind of help with all this major organization we need for this chapter. I really like this one. This gives you a good overview. You can see that innate are non-specific defenses, and adaptive are also called specific defenses. And so you can see again how those are broken down. Now remember, the second type is a specific defense. Here, this is more of a functional system. It helps protect from most bacteria, viruses, transplants, grafts, self cells that have turned against us, which happens with cancer and autoimmune diseases. And it relies on recognizing foreign substances and then communicating with other cells to mount an immune response. Now, since it is called specific, it is literally specific, meaning that these cells have to be programmed and they're specific to only kill whatever they've been programmed for. So this is going to deal with your T and B cells. So the biggest thing here, again, is that they have to be primed. They do not immediately attack. They have to first have an initial exposure to the foreign substance before the body can learn to protect it. Remember, with the nonspecific, it was immediate. Now, let's give an example. So let's say you've got a T cell, and the T cell is going to be programmed to kill strep A. Now, once it gets programmed to kill strep A, it'll wander through your body, killing only strep A. Will it kill strep B? No. It's been programmed to kill only strep A, and so that is what it does. It is specific. Okay, so let's knock out the nonspecific defenses, and then we'll get on into the specifics. So, with the nonspecific, you have the first line of defense, which is your surface membrane barriers. These are all your skin and mucous membranes. So, and everybody always assumes that your immune system is on the inside, but you've got that easy stuff on the outside. That skin we have is amazing. It keeps a lot of stuff from ever even getting in. You've got keratin, which is a physical barrier that's resistant to most weak acids and bases. You've got the acidic pH of the skin secretion inhibits bacteria growth. You've got hydrochloric acid and digestive enzymes in the stomach. So in case you eat those good raw oysters that are full of bacteria, again, hydrochloric acid is going to try to eat most of those so they don't end up in the bloodstream. You've got sebum, which is oil. That's toxic to bacteria. Saliva, lacrimal fluid, which are your tears, they contain lysosomes that actively kill or can digest bacteria. Sticky mucus helps trap pathogens in the respiratory and digestive tracts, and then the cilia works to move it out. So there are a lot of just real simple physical and chemical barriers. Now, your second line of defense, still nonspecific, are cells and chemicals. We'll be talking about phagocytes, natural killer cells, antimicrobial proteins, and inflammation, which will be bringing white blood cells and macrophages to our defense, and then fever. So let's start with phagocytosis. All right, you have something called adherence. A phagocyte recognizes carbohydrates on the microbe surface. It can attach easily when complement proteins and or antibodies coat a foreign substance. Now, so what does that mean? All right, that means that, remember, a phagocytosis means that's cell eating. So what they do is they literally engulf around the bad cell and eat it and break it down. Now, how does it know it's a bad cell? Well, it does so by recognizing those carbohydrates. Okay, all those sugars. It can read those sugars on the surface. And remember, you also have antibodies that are going to be marking bad cells for destruction. So they're going to come in and put a big X on it. And again, the phagocytes will then know it's bad. And this typically works out really good because, again, you've got complement proteins, antibodies. They'll actually coat it and make it sticky, so it's easier to grab hold of and pull in. Pneumococcus has figured this out. It's harder to attach because it has an external capsule made of complex sugars preventing adherence, so it can't grab hold of it, which is why when you have pneumonia, you have to get on antibiotics to clear it out of your system. Your own immune system can't clear it out. And that's another thing that, people don't realize too. They assume that if they have to go get antibiotics, they're sick. And what if you got sick and you didn't have to go get antibiotics? You're still sick. Your immune system saves you from millions and millions of things. But yes, occasionally it does get overwhelmed, or you get stuff like pneumococcus that it can't fight, and then we get on antibiotics to kind of help it out. Now, you have these cytoplasmic extensions that are going to pull in the microbe, forming a phagosome. That phagosome fuses with the lysosomes, and then the enzymes inside the lysosomes degrade or eat the microbe. All right, so in this picture, you can see what a phagocyte looks like, and you can see all those cytoplasmic extensions. They're reaching out and grabbing for that adherence, trying to get those molecules in. And then you can see the phagosome and the lysosomes merging, and then once they eat it, they just get rid of it, spit out the remains, and they move on to the next one. Natural killer cells. These are large granular lymphocytes. Now, we're going to be talking about other lymphocytes like your T and B cells. Okay, this is one of those lymphocytes that did not turn into a T or B cell. Instead, it became a natural killer cell. Now, they're found in your blood and lymph. They lyse and kill a variety of cancer cells and virus-infected cells. They're not picky. Remember, they're nonspecific. They're just going to kill anything. Now, they're not phagocytic. They have direct contact with the target cell and induce apoptosis, which is programmed cell death. These guys are they're like the badasses. They come up to a bad cell, a target cell, and they basically reprogram it to kill itself. I mean, how cool is that? And then, of course, they secrete chemicals that enhance the inflammatory response. So what they do, they find the bad cell, they reprogram it, make it kill itself, and then they basically send out an alarm. Just letting everybody know, I found this guy, there may be some others, you might want to start checking things out. How cool is that? Now, an inflammatory response. This is a response to tissue damage, which includes redness, heat, swelling, and pain. Now, the effects, well, it prevents the spread of damage with clotting factors. It disposes of cell debris and any pathogens. You've got your neutrophils and your monocytes, which are white blood cell types. They squeeze through the blood vessels, which we call diapedesis. We already talked about that in previous chapters. And they go to the damaged area and clean up the debris and set the stage for the repair with a fibrin mesh. Now, there are symptoms like redness, heat, swelling, and pain. All right, so you got the release of chemicals. A damaged cell releases histamines, kinins, prostaglandins, leukotrienes, and complements. This causes the small blood vessels in the injured area to dilate, causing hyperemia or redness and heat. They're opening up so that you can get these massive white blood cells, the neutrophils and monocytes, have to be able to get to the injured area. So they open up that blood vessel so they have plenty of room. Now, the increase of permeability of local capillaries causes exudate to seep from the blood into the tissue spaces. Well, what is exudate? That's a fluid that contains clotting factors and antibodies. If you've ever had like an injury, a wound, and you saw that kind of clear stuff coming up, that's exudate. So it's not bad, it's not infection, it's actually stuff killing the bad stuff. But again, this will cause swelling and then eventually pain because the swelling of the tissue then pushes on the nerve endings, and that's where you get the pain from. All right, so if we're going to mobilize these phagocytes, we've got a couple of steps. First, you have leukocytosis. This is where the injured cells produce leukocytosis-inducing factors that causes neutrophil release from the bone marrow. So essentially, you cut yourself, you get an injured area, immediately you release chemicals to the bone marrow and go, okay, I need you to start amping up, start releasing more of these neutrophils for me. Then you have margination. That's where those new neutrophils then have to travel to the inflamed site, and they stick to the capillary wall, and then they go through diapedesis where they squeeze through the capillary wall, and then you've got chemotaxis. Remember, and we've talked about it before as well. That's where those the injured area is releasing inflammatory chemicals to tell the neutrophils the precise location to go to. All right, so in the picture, again, you have step 1. You've got leukocytosis, where the red bone marrow is going to be making us new neutrophils. Then you've got margination. That's where those neutrophils are going to go to the precise spot on the capillary and stick to the wall. Then you go through diapedesis, where they squeeze through the capillary wall into the tissue. And then finally, you got chemotaxis, where the injured area is releasing chemicals to direct those neutrophils to the injured spot, so they're not just wandering around in the tissues lost. Now, monocytes will follow neutrophils and then develop into macrophages to help. Now, pus is a mixture of dead neutrophils, broken down tissue cells, and living and dead pathogens. An abscess is the pus sac walled off by collagen fibers, so it needs to be surgically drained. Now, this can be caused by a failure to clean out the debris, either by the macrophages or simply by you not keeping the area clean. All right, a next component to the nonspecific defenses is antimicrobial chemicals. All right, you've got complement proteins. There are 20 plasma proteins in the blood that function as complement proteins. All right, you have complement fixation. This is the attachment of the protein to a foreign body, which activates the complement protein. Then you have the membrane attack complexes, which are formed by complement fixation and can punch holes in the cell membrane, amplifying the inflammatory response. All right, complement proteins, I mean, I just explained them, but even this kind of sounds complicated. These are the easiest thing to understand in the immune system. Literally what happens is these complement proteins attach to a foreign body, and then they form these complexes that poke holes in it, and then the cell just oozes out. It's like popping a hole in the balloon. You've killed the balloon. Same thing here. It's actually quite easy. All right, another antimicrobial chemical is going to be interferon proteins. All right, here you got to understand first about viruses. Viruses are not alive. They rely on cell machinery to reproduce. So what happens is the virus has to take over your cell, spit out your cell's DNA, and insert its own RNA, and then our cell makes DNA out of that RNA, and keeps making copies of that virus over and over and over again. So, now that you kind of understand that, let's talk about what the interferons do. All right, once a cell gets infected with a virus, it immediately starts releasing these interferon proteins to the neighboring cells to prevent the virus from infecting them. So what happens? Well, that causes the uninfected cells to produce PKR protein that interferes with virus replication and activates macrophages and natural killer cells. All right, so we're not killing the cell that's been infected with the virus. There's nothing we can do. That cell's done. It's gone. It's gone to the dark side. We're not getting it back. But what we can do is prevent the virus from spreading to our other cells. So that's how we combat viruses. This is also why we can't give you antibiotics for viruses, because if the antibiotic were to attack, what would we tell it to attack? Your own cells? And then how would it know your good cell from the cell that had been hooked by the virus? It doesn't have that capability. So that's why with viruses, there's really not a whole lot we can do. We just tell you to let it work through your system, let your immune system take care of it. Now, in certain viruses like Hep C, herpes, even types of cancer and other viral infections like in organ transplants, where it's a life or death type matter, then we're going to put you on interferons. And typically we do interferon shots. And again, what this is doing is preventing that virus from spreading. All right, so here you can see on the left, this is a cell that's been infected by a virus. And you look to the bottom of the picture, you can see where it releases the interferon proteins to the neighboring cell, which then makes the PKR and blocks the virus from being able to replicate inside that cell. Thus, it's good. It's not going to get taken over. We've prevented the spread. Now, new proteins will also block viruses from replicating, and then again, what else it does is it also activates your B cells and neutrophils, kind of letting them know, hey, we got a pretty active virus going around here. You might want to wake up, start patrolling. Fever. Fever is an abnormally high body temperature. You have pyrogens. Pyrogens are chemicals secreted by white blood cells and macrophages, and they are what cause the body temperature to increase. Now, a mild to moderate fever is essential in helping the immune system because what it causes is the liver and spleen to start gathering iron and zinc. Now, that's important because bacteria need these nutrients to multiply. It also increases the metabolic rates to speed up any repair processes. Now, high fevers are dangerous due to enzymes and proteins being broken down. So if you think back to the chemistry chapters where you talked about proteins denaturing, so they only work at certain temperatures. So when your fever gets too high, then those proteins denature and fall apart, and that's a huge deal. Now, having said that, a couple of things I want to talk about. I love to see these commercials now that tell you, take zinc for your immune system. You should always take zinc. And then you have these people who unknowingly, who have no medical background, go, wow, I get sick a lot. I should take zinc. And then they take zinc and they actually get worse. Now, why? Why would you even want to take zinc? All right, so let's talk about it for a second. Obviously, bacteria use zinc to grow. So if you have a bacterial infection, zinc is the worst possible thing you can take because basically you gave it a food source to grow with. However, if you have a viral infection, zinc actually adds an extra coat to your cells and helps prevent the virus from spreading. Okay, but how do you know what you have? Nobody knows unless you've gone to the doctor and they've told you, yes, indeed, 100% you have a bacterial infection versus a virus or vice versa. So we don't know. So the best thing to do, don't be taking extra zinc or anything else. Let your body take care of it or see a doctor. Don't self-medicate on that. Now, second thing I want to talk about is mild to moderate fever, especially when in children. This is where you need to talk to your pediatrician or your child's doctor and be like, okay, at what temperature do I start giving Tylenol and Motrin? Because what we've seen in the past is parents have a tendency to over-medicate when it comes to fevers. Now, I'm not telling you not to monitor the fever. By all means, monitor it because children can quickly jump from like 100.0 fever to a 104 in a half hour. I mean, if that, virus or bacteria all of a sudden catches up and kicks into high gear, they can easily jump like that without hardly any warning. So you definitely have to monitor it. But if your child's got 100 fever, don't give them Tylenol or Advil. They're okay. Let their body do what it's supposed to. Because if you go ahead and give them something to suppress that fever, then they also stop going to the liver and spleen and stop gathering iron and zinc, and they stop functioning as the immune system. So you have to let that fever go to a certain degree. Again, it's best to talk to your doctor to find out exactly, what temperature maybe. Should you start being more concerned? It's the best thing to do. But remember, a little fever is good. All right. Complement fixation. This involves either A, interferons, B, pyrogens, C, membrane attack complexes, or D, phagocytes. And the answer would be C, membrane attack complexes. Now, the inflammatory response symptoms are primarily due to A, complement proteins, B, T cells, C, dilation of blood vessels, or D, diapedesis of the immune cells. And the answer would be C, dilation of blood vessels. Okay, I'm going to stop here, and then this will end this recording. Remember, you have another half of this chapter to go and a second recording to listen to.